Welcome, fellow veterinary enthusiasts, to a deep dive into the intricate world of veterinary neuroanatomy. Today, we're embarking on a journey through the development of the nervous system, exploring its fascinating origins and the malformations that can sometimes arise. Prepare to be captivated by the wonders of the developing brain and spinal cord. Our story begins with the neural tube, the very foundation of the central nervous system. Imagine a sheet of specialized cells, known as the neurectoderm, nestled on the dorsal side of the developing embryo. This sheet thickens, forming the neural plate, which then gracefully folds inward, creating a groove. The edges of this groove, the neural folds, eventually meet and fuse, transforming the groove into a hollow tube, the neural tube. This pivotal moment marks the genesis of the central nervous system. As the neural tube takes shape, it detaches from the overlying non-neural ectoderm. At the junction of these two ectodermal layers, a remarkable event occurs. A longitudinal column of cells emerges, separating from both the neural tube and the non-neural ectoderm. These are the neural crest cells, bilateral columns that will give rise to a diverse array of structures, from spinal ganglia to the cells of the adrenal medulla. Closure of the neural tube is a carefully orchestrated process, beginning at the level where the rhombencephalon will develop and progressing both rostrally, towards the head, and caudally, towards the tail. The caudal closure primarily forms the spinal cord. In the developing brain, closure may begin at multiple sites, with the most rostral opening, known as the rostral neuropore. The spinal cord extends from the caudal end of the closed neural tube as a column of neuroepithelial cells grows caudally between the notochord and skin ectoderm. Cavitation within this column creates the neural canal, ultimately forming the caudal, sacral, and some lumbar spinal cord segments. An opening may persist at the caudal end of the neural tube, connecting with the subarachnoid space at the conus medullaris. The rostral end of the neural tube undergoes rapid development, forming three primary vesicles: the prosencephalon, forebrain, the mesencephalon, midbrain, and the rhombencephalon, hindbrain. Early in development, the prosencephalon sprouts lateral enlargements called optic vesicles, which interact with the overlying skin ectoderm to initiate eye development. Later, two additional swellings emerge from the rostral prosencephalon, growing laterally and dorsally. These telencephalic vesicles eventually overgrow the original vesicular system, forming the cerebral hemispheres. The remaining portion of the prosencephalon becomes the diencephalon, retaining the optic vesicles. The neural canal within the diencephalon forms the third ventricle, which communicates with the lateral ventricles, first and second ventricles, of the telencephalon via the interventricular foramina. Within the diencephalon, the thalamus and hypothalamus develop, and the neurohypophysis emerges as a ventral outgrowth. The cerebral cortex and basal nuclei develop within the telencephalon. The neural canal of the mesencephalon narrows to form the mesencephalic aqueduct. From the rostral rhombencephalon, the cerebellum, or dorsal metencephalon, develops dorsally. The remaining ventral metencephalon becomes the pons. The caudal rhombencephalon forms the myelencephalon, or medulla oblongata. The fourth ventricle represents the lumen of the neural canal within the rhombencephalon. It communicates with the surrounding meningeal spaces via openings in its wall, the lateral apertures. The neural canal continues caudally as the central canal of the spinal cord.
Now, let's zoom in on the microscopic level to witness the remarkable process of cell differentiation within the neural tube. Initially, the cells within the neural tube wall, known as neuroepithelial or neurectodermal cells, are arranged in a pseudostratified layer. Each cell spans the entire width of the neural tube, with nuclei located at various levels. These cells are actively dividing, increasing the thickness of the tube wall. The nuclei migrate within the cytoplasm, their position determined by the cell's stage of mitosis. During interphase, the nuclei reside on the external surface of the neural tube, where DNA duplication occurs. As the nucleus enters mitosis, it migrates to the luminal surface of the neural canal, where cell division is completed. The two daughter cells then extend their cytoplasm back to the external surface, and their nuclei migrate back to the periphery. Here, the cells either undergo another round of mitosis or begin to differentiate. Differentiation occurs at the external surface of the neural tube. A new layer of differentiating cells appears on the outer surface of the actively dividing layer. The mitotic layer is called the germinal layer, and the differentiating cells form the mantle layer. The differentiating cells give rise to two main types: immature neurons and spongioblasts. Immature neurons, the primary parenchymal cells of the nervous system, grow extensively to become mature, functioning neurons. Spongioblasts are the progenitors of the neuroglia, the supporting cells of the nervous system. Astrocytes and oligodendrocytes are derived from spongioblasts, while microglia originate from mesodermal cells, monocytes, that enter the nervous system from the blood supply. As neurons and spongioblasts differentiate and grow, the neural tube organizes into 3 concentric layers. Adjacent to the lumen lies the germinal layer, where neuroepithelial cells proliferate. This mitotic activity eventually ceases, leaving a single layer of cells called ependymal cells, which line the entire neural tube lumen, including the ventricular system and central canal. Peripheral to the germinal layer is the thick mantle layer, composed of differentiating neurons and spongioblasts. This layer becomes the gray matter of the spinal cord, brainstem nuclei, cerebellar cortex and nuclei, and the basal nuclei and cerebral cortex. The outermost layer is the marginal layer, primarily composed of growing axonal processes from the mantle layer neurons. These axons will be myelinated by oligodendroglial cells, forming the white matter tracts. In the developing spinal cord, the neural tube exhibits a clear symmetry. A longitudinal groove, the sulcus limitans, appears in the lateral wall of the neural canal, dividing it into dorsal and ventral portions, the alar plate and the basal plate, respectively. Functionally, the alar plate is primarily concerned with sensory systems, while the basal plate is associated with motor systems. The ventral growth of the basal layers leaves a separation, the ventral median fissure. The marginal layers of the alar plates fuse on the median plane, forming the dorsal median septum. This midline growth reduces the neural canal to the small central canal, lined by ependymal cells. The mantle layer of the alar plate becomes the dorsal gray horn, while that of the basal plate becomes the ventral gray horn. The mantle zone at the sulcus limitans forms the lateral gray horn. Within the mantle layer of each plate, neurons are further organized into functional columns. General visceral afferent and efferent neurons are located adjacent to each other on either side of the dorsal plane through the sulcus limitans. General somatic afferent and proprioceptive neuronal columns are located dorsally in the alar plate, and the general somatic efferent column is located ventrally in the basal plate. The size of each spinal cord segment depends on the volume of tissue it innervates. At the levels of the limbs, the spinal cord segments responsible for their innervation are enlarged, forming the cervical and lumbosacral intumescences. The survival of a developing neuron in the peripheral nervous system depends on its successful innervation of a muscle cell or the formation of a peripheral receptor. Lack of innervation leads to neuronal degeneration. In the basal plate mantle layer, general somatic efferent neurons located medially innervate axial skeletal muscles, while those located laterally innervate appendicular skeletal muscles. Axons of the basal plate neurons grow through the marginal layer and out of the neural tube, forming the ventral root and part of the spinal nerve.
Let us now consider neurogenesis, the creation of new neurons. For a long time, it was believed that once a primitive neuron differentiated in the neural tube mantle layer, it would not undergo mitosis again and would persist for the animal's life. The premise was that neurogenesis ceased once the central nervous system was formed. However, research has revealed that selective postnatal neurogenesis occurs throughout the life of the animal. In the embryonic neural tube, neuroepithelial cells give rise to radial glia. These radial glia guide developing neurons to their destinations and can differentiate into neurons, astrocytes, oligodendrocytes, and ependymal cells. After development, undifferentiated radial glial cells populate the subventricular zone, especially near the lateral ventricle of the frontal lobe. Here, they function as neuronal stem cells, providing a source of astrocytes and oligodendrocytes. Adult neurogenesis occurs primarily in the rostral cerebral subventricular zone, where new neurons populate the olfactory bulb, and in the subgranular zone of the hippocampal dentate gyrus, where they replace granule neurons. The function of these adult-born cells is still under investigation. These subventricular zone adult stem cells are thought to be the origin of some gliomas and may play a role in plasticity after injury. Researchers hope to harvest these cells for cell replacement after central nervous system injury or neurodegenerative diseases. As we journey from the spinal cord to the brainstem, we encounter the medulla oblongata, or simply, the medulla. The medulla's development is a slight modification of the spinal cords. The narrow mid-dorsal roof plate of the neural tube stretches extensively instead of being obliterated by the alar plate and marginal tissue. Imagine grasping the midline roof plate and pulling your hands apart sideways. This stretches the roof plate into a thin layer, displacing the alar and basal plates laterally and ventrally. This enlargement forms the fourth ventricle, covered dorsally by a single layer of ependymal cells. The sulcus limitans on the ventrolateral wall of the fourth ventricle divides the medulla into basal and alar plates, with the same functional significance as in the spinal cord. Throughout the brainstem, the mantle layer breaks up into nuclei, collections of neuronal cell bodies with a shared purpose. These nuclei are interspersed with neuronal processes, and some are more distinct than others. The functional columns found in the spinal cord have similar locations in the brainstem. Additionally, the medulla contains neurons organized into functional groups unique to cranial nerves. Cranial nerves 6 through 12 and the trapezoid body are located within the medulla. The rostral border of the medulla is the caudal border of the pontine transverse fibers. Cranial nerves 6, 7, 9, 10, 11, and 12 contain general somatic efferent neurons. The medullary nuclei of cranial nerves 6 and 12 are located in an interrupted column along the median plane next to the fourth ventricle. The hypoglossal nucleus is very long. The general somatic efferent neuronal cell bodies of the facial nerve migrate ventrolaterally to form the facial nucleus. As a result, axons leaving the facial nucleus initially course dorsomedially before turning ventrolaterally to exit the medulla as the facial nerve. Cranial nerves 9, 10, and 11 also undergo ventrolateral migration, accumulating in the nucleus ambiguous. The sensory components of cranial nerves associated with the medulla arise primarily from primitive neurons from the neural crest cells, with some from ectodermal cells that proliferate from branchial arch ectoderm, called cranial placodes. Neural crest cells form the sensory ganglia of cranial nerve 7, 9, and 10, associated with general visceral afferent and special visceral afferent functions. Ectodermal cells from the otic placode form the sensory ganglion of cranial nerve 8, associated with special proprioception for vestibular function and special somatic afferent function for hearing. These cranial nerve 8 ganglia are located in the inner ear within the petrosal portion of the temporal bone. Their axons synapse on cell bodies in the alar plate region of the medulla. The leptomeninges, which surround the developing central nervous system, arise from neural crest cells and adjacent mesodermal cells. These meninges contain the blood vessels that supply the central nervous system and the roots of cranial and spinal nerves. The roof plate of ependymal cells, covering the fourth ventricle, and the thin layer of vascularized pia associated with it, form the tela choroidea of the fourth ventricle. Capillary blood vessels in this tela choroidea proliferate, forming two longitudinal rows of a dense capillary bed. The adjacent ependymal cells enlarge into cuboidal cells, and the entire structure protrudes into the fourth ventricle lumen, forming the choroid plexus of the fourth ventricle. The choroid plexus of the fourth ventricle comprises two sagittal lines extending rostrally from the caudal part of the fourth ventricle to the level of the cerebellar peduncles, where each plexus turns laterally. At this point, the lateral aperture develops in the medullary roof plate, connecting the fourth ventricle lumen with the subarachnoid space. The choroid plexus protrudes through the lateral aperture, visible at the cerebellomedullary angle. The choroid plexus is a major site of cerebrospinal fluid formation. In domestic animals, the lateral aperture is the only communication between the ventricular system and the subarachnoid space. Primates have an additional median aperture.
Moving rostrally, we encounter the metencephalon, which gives rise to the cerebellum and pons. Cranial nerve 5, the trigeminal nerve, is associated with this brainstem segment. Its motor neurons arise in the basal plate mantle layer and migrate a short distance ventrolaterally into the pons, forming the motor nucleus. These neurons function in the general somatic efferent system and innervate the muscles of mastication. The sensory neurons of cranial nerve 5 are derived from neural crest cells that form the trigeminal ganglion. Most are general somatic afferent neurons with dendritic zones spread over the head and the mucosa of the respiratory and digestive systems within the head. A smaller component comprises general proprioceptive neurons for head muscles and joints. These sensory neurons greatly outnumber the motor neurons. Their axons enter the alar plate region of the metencephalon, spreading rostrally and caudally, forming the spinal tract of the trigeminal nerve in the pons and medulla. These axons terminate in telodendria at synapses in alar plate neurons, forming the sensory pontine nucleus in the pons and the nucleus of the spinal tract in the medulla. The cerebellum, or dorsal metencephalon, is formed from the proliferation of germinal epithelial cells of the alar plate, forming the rhombic lip. This dorsolateral growth replaces the roof plate of the fourth ventricle, making the cerebellum part of the dorsal boundary of the fourth ventricle in the metencephalon. The ventral metencephalon is the pons. A ventral migration of alar plate mantle layer neurons forms the pontine nucleus. The axons of these neurons cross the midline and course dorsally into the cerebellum, forming the transverse fibers of the pons, which demarcate the ventral surface of the pons and continue into the middle cerebellar peduncle. Further rostrally lies the mesencephalon, or midbrain. Symmetric proliferation of the neural tube walls in the mesencephalon reduces the neural canal to a narrow tube, the mesencephalic aqueduct. Cranial nerves 3, oculomotor, and 4, trochlear, are associated with the midbrain. These primarily contain general somatic efferent neurons that innervate extraocular muscles. Their cell bodies are in nuclei derived from the basal plate mantle layer and remain adjacent to the median plane ventral to the aqueduct. The parasympathetic nucleus of the oculomotor nerve, containing preganglionic parasympathetic innervation to the constrictor muscle of the iris, is just rostral to the oculomotor nucleus. The alar plate proliferates dorsally to form the tectum, divided into paired rostral and caudal colliculi, collectively known as the corpora quadrigemina, associated with visual and auditory reflex functions, respectively. The crus cerebri on the ventral surface of the midbrain results from the caudal growth of axons from telencephalic projection neuronal cell bodies, continuous from the internal capsule in the diencephalon. Our journey concludes in the diencephalon and telencephalon, the most rostral parts of the developing brain. Rostral to the mesencephalon, the sulcus limitans is no longer evident. The diencephalon and telencephalon are considered developments of the alar plate. Symmetric development of the lateral walls of the neural tube in the diencephalon reduces the neural canal to a vertical slit on the median plane, the third ventricle. Adhesion of the developing thalamus in the center forms the interthalamic adhesion and separates the third ventricle into dorsal and ventral components. These converge caudally at the mesencephalic aqueduct and rostrally at the interventricular foramina, which connect to each telencephalic lateral ventricle. Proliferation of neural tube epithelial cells does not occur on the dorsal median plane of the diencephalon, leaving a single cell-thick roof plate where a small choroid plexus develops in two parallel lines, similar to those in the medulla. These are continuous with the choroid plexus that develops from the roof plate in each lateral ventricle. A plethora of nuclei forms from the mantle layer in the diencephalon, forming the thalamencephalon, hypothalamus, and subthalamus. The thalamencephalon consists of the thalamus, metathalamus, and epithalamus. The hypothalamus includes nuclei on the sides and floor of the ventral portion of the third ventricle. The subthalamic nuclei are located ventrolaterally in the diencephalon. A ventral outgrowth of the hypothalamus, including an extension of the third ventricle, forms the neurohypophysis and hypophyseal cavity, respectively. The neurohypophysis becomes associated with a dorsal extension of the oral ectoderm, the hypophyseal pouch, to form the hypophysis or pituitary gland. The optic vesicles, which initially grew out of the prosencephalon, form the neural layer of the eye and optic nerves associated with the diencephalon. The axons that grow caudally from the retina in the optic nerve form the optic tracts of the diencephalon, terminating in a thalamic nuclear area. These optic nerves form cranial nerve 2, the special somatic afferent neurons of the visual system. Each telencephalon forms a cerebral hemisphere, and the two hemispheres form the cerebrum, connected by the corpus callosum. The lamina terminalis of the diencephalon is the rostral boundary of the brainstem, located on the median plane between the telencephalic outgrowths. The optic chiasm is located at the ventral portion of this lamina, and the rostral commissure develops and remains in this lamina. At one aspect of the medial wall of the telencephalic vesicle, the neuroepithelial layer of the neural tube does not proliferate, remaining a single layer of ependymal cells. This telencephalic roof plate is attached to the crus of the hippocampal fornix on one side and the stria terminalis on the other. The choroid plexus of each lateral ventricle develops in this roof plate. At each interventricular foramen, each lateral ventricular choroid plexus is continuous with the choroid plexus of the third ventricle. Extensive development of projection axons occurs from diencephalic thalamic neurons to the cerebrum and telencephalic neurons to the brainstem, forming the thick layer of myelinated axons, the white matter between the diencephalon and telencephalon, known as the internal capsule. The neuronal cell bodies are located on the external surface of the telencephalon, forming the cerebral cortex, or deep to the surface in subcortical basal nuclei. The cerebral cortex is divided into the archipallium, the paleopallium, and the neopallium. Association, projection, and commissural axons form three groups of telencephalic neuron processes. All three groups of commissural axons initially develop in the lamina terminalis. In the telencephalon, the neural tube germinal layer is ultimately replaced by the ependyma of the lateral ventricle. Except for the area of the basal nuclei, the mantle and marginal layers reverse their positions. This is because newly formed neurons migrate to the neural tube surface to form the cerebral cortex, and their axons grow centrally, forming the white matter.
Now, let us turn our attention to the malformations that can arise during the development of the nervous system. One such malformation is hydrocephalus, an enlargement of the ventricular system, usually most evident in the lateral ventricles. The most common cause is interference with cerebrospinal fluid flow from the ventricles into the subarachnoid space, resulting in increased cerebrospinal fluid pressure. This hypertensive hydrocephalus causes extensive degeneration of the telencephalon, especially the neopallium. Another is hypoplasia of the prosencephalon, also known as cerebral aplasia or anencephaly. Calves with this malformation are born with failure of the telencephalic vesicles to develop, associated with a small opening in the calvaria on the midline where fluid emerges. The brainstem and cerebellum are present but reduced in size and abnormally shaped. The eyes are well developed, and all cranial nerves except the olfactory neurons are present. In humans, this is called anencephaly. The cause of this malformation in animals is unknown but may result from folic acid deficiency in the mother early in development. Affected calves are usually unable to get up at birth and are blind. A meningocele or meningoencephalocele presents as a large skin-covered swelling outside the calvaria, usually on the midline. A meningoencephalocele, which contains brain tissue in the swelling, is more common. The calvarial defect is called cranioschisis or cranium bifidum. This is usually on the midline and can be at any level along the calvaria. In large meningoencephaloceles, the protruding cerebral hemispheres are usually very thin and fluid-filled because of dilation of their lateral ventricles. The cause is usually unknown. It is seen in all species of domestic animal, but may be more common in pigs. A very large meningoencephalocele occurs as part of an inherited craniofacial malformation in Burmese cats. In addition to the large meningoencephaloceles, these kittens have significant facial malformations, with shortened jaws and nasal areas, and absent olfactory bulbs or peduncles. These facial deformities are the result of abnormal cranial neural crest development. Exencephaly is defined as brain tissue protruding out of the cranial cavity and not covered by skin. Both exencephaly and meningoencephalocele require a cranioschisis, the opening in the calvaria, where the bones have not fused. A lipomeningocele is a meningocele with a large amount of associated fat. It may occur along the midline of the calvaria or vertebral column through a cranioschisis or spina bifida, respectively. With no associated neural tube malformation, no neurologic signs are present. Duplication of the prosencephalon occurs most commonly in calves and is associated with varying degrees of facial duplication, called diprosopis or dicephalus. The brainstem usually becomes single somewhere in the mesencephalon. This is a partial dicephalus. These calves are usually born alive but are recumbent and unable to stand. Holoprosencephaly occurs with the cyclopean malformation. In lambs, this occurs when their dams are exposed to Veratrum californicum during pregnancy. This results in a failure of the visual area to divide into two fields. A single midline optic vesicle develops, the absence of any nasal portion of the respiratory system and olfactory system, and the presence of a proboscis dorsal to the eye. The interference with prosencephalic development results in only a single midline telencephalic vesicle, holoprosencephaly, that grows caudally over the brainstem. Hydranencephaly is another term for this cerebral malformation because development of the olfactory system fails to occur. Hydranencephaly is a cerebral abnormality in which the neopallium is reduced to a thin, nearly transparent pial-glial membrane lining the expanded lateral ventricle. The loss of neopallial parenchyma represents both aplasia resulting from viral destruction of germinal layer cells and atrophy resulting from the viral destruction of already differentiated neopallial neurons. Usually, the olfactory paleopallium, the archipallium, and the basal nuclei are spared. Bluetongue virus has been reported to produce this lesion in cattle and sheep. The nature of the lesions depends on the gestational age at which infection of the fetus occurs. In lambs, necrosis of the germinal cells prevents them from contributing to the formation of the neopallium. Hydranencephaly occurs in calves exposed in utero to bluetongue virus around 125 days of their in utero development. Acobane virus has also been associated with hydranencephaly in ruminants. Lissencephaly means a smooth brain without evidence of cerebrocortical folding to produce gyri and sulci. The cerebral cortex is thickened because of the abnormal distribution of neuronal cell bodies. The malformation presumably results from impaired neuronal migration, and an inherited genetic abnormality is presumed in some breeds. Clinical signs include difficulty in learning, abnormal behavior, and occasionally slow postural reactions and poor menace responses. Complex malformation of calves has been described as Arnold-Chiari malformation. These calves are born alert and with vision but are recumbent and unable to coordinate their neck, trunk, and limb functions. A sacrocaudal spina bifida is present, with a meningomyelocele, a malformed tail, and an associated loss of tone and reflexes in the anus and tail. Occipital bone malformation and syringohydromyelia occur most commonly in small breed dogs. A dilation of the central canal is hydromyelia, and a cavitation of the spinal cord parenchyma is syringomyelia. The cause of the herniation is presumed to be a caudal cranial fossa that is too small for the volume of brain tissue it contains. Clinical signs vary in onset from a few months to the early adult years and most commonly relate to the cervical spinal cord. The most common clinical sign is discomfort. Diagnosis is made with magnetic resonance imaging. Calvarial ossification variations are often mistakenly related to neurologic disorders. One involves the occipital bone at the foramen magnum and the other the frontoparietal suture. A dorsal notch or extension of the foramen magnum has been referred to as occipital dysplasia. Incomplete ossification at the frontoparietal suture is referred to as a molera or fontanelle. Several important features regarding spinal cord malformations must be emphasized. The clinical signs will be present at birth or when the newborn animal first tries to walk, and they will not progress. Spinal cord malformations are most common in calves and most commonly occur in the thoracolumbar segments. A very common lesion is duplication of the spinal cord, called diplomyelia. Less commonly, two spinal cords are present, each with its own meningeal sheath and each in its own vertebral canal, separated by a bony partition, called diastematomyelia. Regardless of the nature of the thoracolumbar spinal cord malformation, if the animal is able to walk, it is common for the pelvic limbs to be used simultaneously in a bunny-hopping fashion. Spina bifida and meningomyelocele are two distinct malformations that are often confused. Spina bifida refers to the failure of the closure of the dorsal aspect of the vertebral foramen of one or a few vertebrae. A meningomyelocele is a protrusion of the meninges and associated nervous tissue outside the vertebral canal. A sacrocaudal spina bifida and meningomyelocele are most common in the Manx breed of cat and in English bulldogs. The Manx cat is known for the absence of its tail, which, in fact, is the trait that is selectively bred for. In dogs, the most familiar and well-studied spinal cord malformation is spinal dysraphism, which is inherited in the Weimaraner breed. Spinal dysraphism is defined as an incomplete closure of a raphe or defective fusion of the neural tube. The clinical signs are first observed as these dogs begin to walk, when they consistently exhibit a simultaneous gait with the pelvic limbs.
Understanding the pathogenesis of malformations is crucial for preventing the birth of further affected animals. Confirmation of the malformation's morphology, usually requiring a necropsy diagnosis, is essential. If the disorder is inherited, a confirmed diagnosis is needed before making judgments about the parents. Recent advancements in gene code studies have led to blood tests that identify affected and carrier animals, providing the opportunity to significantly decrease and even eliminate many of these malformations. And that concludes our exploration of the developing nervous system and its malformations. It's a field brimming with complexity, offering endless opportunities for discovery and innovation. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.